Well, thank you, everybody. We're running a little bit late, so try to make it as interesting. And relevant as possible. I'll cover a little bit of what Klaus has covered in that video, and again, he'll answer questions directly by email. But if you do have any questions on his particular presentation, I might be able to help there. It's always a pleasure to come to Spain. I thank Juan and his team. They're always incredibly hospitable. They always organise beautiful weather for me, which is helpful. And I will do my best to speak slowly, so if you have any questions, it is supposed to be a workshop, so please interrupt at any time. So, my first question, who would like to work for Klaus Wiemer? As you can tell by the way he showed about making dishes, he's very particular about how he runs his labs. And he's very modest, of course, and says we're quite happy with a 50, 60, 70% pregnancy rate. I mean, who wouldn't be? So I think I take from what he presented that really now in Europe we have no choice but to use global LP if we want to use global media. The LGPS system, which was brought in from the very beginning, is no longer available in Europe. It will never get a CE mark because of the nature of how it's produced. So we had to find an alternative. And fortunately, the data that we've heard earlier, and hopefully what Klaus has presented, has convinced you at least that LP works as well as LGPS, and hopefully will work as well in your labs with any other media that you potentially want to use. I would also like to thank the presenters from this morning for putting my heart in the right place and not giving me any palpitations. My role within the Medical Affairs Department and at Cooper Surgical is one where we want to get the best out of the media and every other piece of equipment or consumable that we manufacture. And it's part of our role to make sure that you guys get the best out of it. And so when you have to sit and listen to people presenting on what they've used of yours, you hope that it's going to give you good results. And mostly because we certainly don't want to do any harm to any of the patients that have obviously been using these media and many other things that we produce. So thankfully everything seemed okay. It's exactly what I expected to hear. It's never going to be universally better than what people currently use in an environment where it's always going to have a bias against it, because people are using a media, whatever that might be, and this is the test media, and so consequently they've had a lot of experience with one and very little with another, and so you always expect a little bit. And I can show you in the presentation some presentations or some publications, and they rarely show huge differences in outcome because they're not powered to. And even Klaus has said in his study, the numbers are not there to say one way or another whether one's better than the other. They can basically say more or less they're the same, but we'd need many, many more patients to go through treatment and go through this protocol before we could say one was better than the other. So ultimately, this is what I'm hopefully going to cover. As I say, I will speed up a little bit because it's getting towards 4 o'clock and I can tell people have filled their brains. So, global media. Global media, be it culture media, fertilization media, whatever, has been designed to be a family of media. This was where the principles came from at the beginning. So whichever one of them that you use, they all have the same chemical backbone, and they only change in their formulation what's needed for that particular function. So, for example, fertilization media needs higher glucose concentration for sperm survival and cumulus cell health. So it has higher glucose concentration than the culture media, but everything else is the same. And it's the same for the others. Whatever their function is, HEPES replaces bicarbonate, but everything else is the same. And we want to do it for one reason alone, is that we want to keep the embryos stress-free throughout their development, and this seems to help. And we know that that's the case. But I do want to reiterate before I get into the media that anything that we use in the lab is part of a media or a culture system, and the culture media is only one part. And if we don't get the other parts right, the culture media cannot perform to its optimum. And so this is just some of the things that we might think about that could impact our treatment and our success. And teamwork is obviously one of them. And again, one of the good things about coming to Spain is that the embryologists I meet in Spain are all generally very well informed and have good labs and good success rates. That's not the case worldwide, and you'd be surprised and horrified at some of the things that I do see, and one of these days I may write a memoir and share them all with you. But teamwork's incredibly important. Who would like to work in a lab doing 3,000 cycles of IVF per month? Okay, so we now have several clinics in China doing 35,000 cycles per year. I don't know anybody in Europe that would want to work in that environment. There is no teamwork, it's a factory, there's no quality control. How they don't mix up gametes is very hard to say. So, we're lucky in Europe, we have strong regulation here, so we have to do things the right way. And so these are some of the practical aspects. Quality control is extremely important, and again, if we don't get this right to maintain things on a regular basis as the way they should be, we will never get the best results. Okay, so bear that in mind. We'll talk a little bit about it later. You heard this morning, my colleagues did a great job in explaining some of this to you in Spanish, so you've heard it in Spanish and now in English. So the two philosophies that have come around about designing media are either you use a single-step media, i.e. let the embryo choose what it wants from its environment, or you use something that you design based on observation. Now, actually, if you look at defined media for anything in science, it will tell you that you shouldn't use physiological observations to determine it because they are variable, and you should actually use just observation to determine what the optimal media is. And so you can make your own judgment, but we know they both have their limitations. The sequential media had its limitations by the variability of observations in the various things that they measured. And single-step media, so simplex optimization media as it was, has its limitations because it was done in a mouse model. We can't do this in the human, so we had to extrapolate from the mouse. And as everybody knows, mouse and humans are not necessarily the same. Some people could argue differently, but we are not the same, and we have some distinct advantages, not least of which, if you put cleavage stage embryos back in any other species other than the human, you get pregnancies. So thank God for that, otherwise we wouldn't all be sitting here today working in this field. So again, that approach leads us to the single step or the sequential media, and again, I don't deny at all most of the commercially available media. If you use it at its optimal, you will get some background results. We know that. Whether you get optimal results depends on many other factors. So the questions that we ask, about the concentrations of the components that we saw, this is really only is biased against sequential media, but as I said, single-step media does have its drawbacks in how it was developed. But ultimately, is anything normal, natural? And I like this next little cartoon because it tells you everything you need to know about what we do in the lab. So on the left is a very dynamic in vivo environment. The embryo development is not in a big pool of liquid. It's in very close contact with the walls of the fallopian tube. It has interactions with all of that. The analogy that we use on the right, that what we do in the lab, is putting a pea in a swimming pool. Okay, this is not normal. And so either sequential or single-step media are never going to be normal. They're very far from it. So we need to bear that in mind. We can't mimic what happens in vivo. Whether we ever will be able to, whether microfluidics will come along at some point and help, who knows? But at the moment, we're a long way. So I don't think you can say sequential or single-step media are normal. They're a long way from what you see in vivo. So bear that in mind as well. So the history of global. And Klaus's name's on here in 2002. So it started off as simplex optimization media. John Biggers, who was the doyen of culture media, was involved from the very beginning. It became KSOM for human work, so increased potassium concentration. Then Klaus and John Biggers introduced amino acids. If you put the same concentration of amino acids you put into animal cell culture, human embryos die. So they thought, well, what should we do? And they literally just halved the concentration of amino acids, essential and non-essential, and suddenly human embryos are quite happy. Then they changed the glutamine amino acid to a dipeptide, which became the initial global, and it became alanine glutamine, and then it was changed to glycyl glutamine, and that remains the same today. So culture media itself is not actually that complicated. Some of you have probably heard this, when I started as an embryologist, I was told in my training that the culture media was the most important thing that we had in the lab, and therefore it would be the last thing I was taught to make. And when I was actually shown how to make media, I was horrified. We had a number of bottles that we lined up. We added water, salt solutions, protein, antibiotics. We inverted the bottle a couple of times, and then the embryologist held it up to the light. And I said, "What are you doing?" He said, "I'm checking the pH." I said, "What?" I said, "This is a solution we're going to inject into the patients." He said, "Your eyes are very, very sensitive to colour, and this is the way we've always done it." So I said to him, "So what if the pH is incorrect? What do we do?" "Oh, well, we have 1 molar sodium hydroxide, and we have 1 molar hydrochloric acid, and depending on whether it's too acid or too basic, we add them dropwise. So as you all can imagine, if you want it at 12 o'clock, you start here. So you add 1 and you go past. So then you add the other and you go back the other way, and you end up..." So that was bottle number 1. So bottle number 2 then doesn't need any alteration. And so I said, so we've made 10 bottles of media. They're all different because they've all had different amounts of things added to them. And we're going to inject these into a patient, and we've checked the pH by eye. And he said, "Yeah, but that's the way we've always done it." I said, "So if somebody from outside came to the lab and asked us how we made the media, you'd be happy to tell them that one of the critical things that we do, we just hold it up and say, well, it's about 7.2. Amazing. And we've come a long way from that. Unfortunately, the hangover from it is that now, should anything untoward happen in the embryology lab, the media gets the blame. So if your results dip at all, it's always who made it, or where's the media, who's it, what lot is it, where did it come from? It must be the media. And unfortunately, we know nowadays it's probably not, but it still gets the blame. So it's a very simple solution. Salts, a buffer, some energy sources, amino acids, EDTA for chelation, and that's 1 dipeptide to stop ammonia production, antibiotic, and phenol red as a pH indicator. And then just to reiterate, this is the global family. And I like this slide because it doesn't matter whether it's global LP, global HP, LGPS, all applies the same. So the protein concentration in each of those will be different. But if you look down, the sodium chloride concentration across every product in the range is exactly the same. The potassium chloride is the same. And then it only varies where it needs to. So where you look at buffer, where it needs to work in a CO2 environment, bicarbonate, where it needs to work outside the incubator, HEPs. That's as simple as that. So we try to make it so that when the embryos are moved from one solution to another, they are as stress-free as possible. So again, just to reiterate, they're all the same. Remarkably, in the very beginning, we used to use BSA, bovine serum albumin, in human embryology, would you believe? Fortunately, we don't anymore. So, also mentioned this morning, oil is absolutely critical. The number of times I talk to embryologists about why they're cutting down on the amount of oil that they use to save a little bit of money. And as we've moved from doing day 2 transfers, which is what I started with, where we hid a lot of the errors that were going to occur in the culture media, we've now moved to day 5, day 6. Oil has become even more critical. If you don't use enough oil or use too smaller drops over the time of culture, you will change the environment that the embryos are in without doubt. And the other thing, again, is if people take account of the viscosity of the oil, this has an impact on the equilibration time for the dishes. You need at least 12 hours, even with the light oil or paraffin oil. And if you get to lifeguard oil, which is very viscous, then you need even longer. Klaus said 19 hours. He's always at the extreme of everything. 19 hours is a long time, but it indicates to get fully equilibrated dishes, we need to make sure they're in the incubator for a long time. So this is, again, just to illustrate that, and this is very nicely indicated, because if we look at our drop, this is what I see embryologists doing. I see them holding the dishes up and looking. I say, what are you doing? Oh, I'm checking the oil is covering the drop. And say, but if you do it that way, so it's literally just covering, you will end up, without doubt, in extended culture with loss of water, and therefore you'll have increases in the salt concentrations, the pH will change, the osmolality will change, everything. So drop size is important, oil is very important, and as we heard earlier, mostly because of volatile organic compounds. Not all of us work in the best labs in terms of air quality, and that again varies around the world. So we need to make sure that we use enough oil and our drops are big enough so that we have a good volume to surface area ratio, and again we don't get evaporation. Jason Swain did a paper, his name was mentioned a few times this morning, where he did different types of dish making, and as Klaus did with the GPS dishes, he recommends that you put drops in your dish, you put oil over the top, then you take the drops out and you put new media in, and this way you minimize any changes that you might get. It's a pain in the neck to do it, particularly if you have a busy clinic making dishes that way, but that's the way he recommends to have the least change over time. So a single step media can be used to culture from zygote to blastocyst without question. There's enough data out there, but you need to be careful that you have a good system in your lab to allow that to happen. And changing media, whether it be the same media or a different media as in sequential on day 3, is a stress on the embryo, and we need to minimize it as much as possible, as is opening and closing the incubator doors. So if you're going to introduce any media into your lab, this is for global, but it could be any, you need to have a plan of how you're going to do it and make sure that you do it right so that nobody is compromised in any way. And really we can't get a really conclusive answer in the time frame that is commercially viable. So no, I don't believe any company is going to be able to come to an embryologist who says, yes, I want to trial your media, and I need to do 1,000 patients in your arm with your media and 1,000 patients with the media we currently use, and that will help me decide. It's just not feasible to do that. So we recommend initially 30 to 40 patients with the test media, gives an indication of whether it's going to work in your system. And then beyond that, we can talk about how we refine it. But you can never get a truly positive answer in terms of significant results with a relatively small sample number. It's recommended that you do a prospective randomized trial. Some places do, and it was nice to hear somebody did this morning. Most places don't do that. They do an evaluation rather than a trial, partly because if you call it a trial in many countries, you then have to get ethical approval, and that again adds to the complication of doing it. So we talked about optimal culture conditions. I stress time and time again to people, so mostly they listen, sometimes they don't. Every media is different. So you cannot assume that a test media will work in exactly the same way as your current media. And the main consideration for that is bicarbonate concentration. And we had a discussion over lunch between Sage 1-step and Global. Now, Sage 1-step has bicarbonate concentration about 50 millimoles per litre, and Global has about 19.5. So at the same CO2 concentration in the same incubator, they will not give you the same pH. And therefore, depending on what the optimal pH is, they may or may not be working at their best, and you need to bear that in mind. And this graph on the right shows different media, different CO2 concentrations, and different pHs. And as you can see, they're almost all different. So even in the same environment, you do get differences, and you need to bear that in mind. So we recommend doing sibling oocyte trials or sibling zygote trials. Again, some people will do it, some people won't, depending on their workload. But in some way, you need to assess whether one media is going to work better in your hands than the one that you currently use. And certainly as far as Global is concerned, over the time period that we've had it in the market, we have worked out what the optimal pH is. If you ask any other commercial company what the optimal pH of their media should be, they'll tell you between 7.2 and 7.4. Now, we know for global that that's not true. It will work within that range, but if you want it to work optimally, it should be below 7.3 and should ideally be somewhere between 7.25 and 7.29. Now, that's a much smaller range than 7.2 to 7.4, and it has its implications. So you need to test it in your system. You need to make sure that you use an incubator dedicated to the trial media or the evaluating media, and it should be the same as one that you're testing against. I was in a lab last week where the CO2 concentration was set the same for each of their incubators in the lab, including a jerry, some benchtop, some box incubators, and every one gave different pH readings, even though they were all set at the same CO2. So it is incubator dependent as well, and environment and everything else does play a part. So bear that in mind. If you're going to compare two media, they should be given equal opportunity to work at their best and not compromise them. Again, the worst thing that can happen, and this has happened in some quite famous labs, they decide on two media. One works at relatively high pH and one works at relatively low pH, so they choose somewhere in the middle. And now all that does is it compromises both. So you'll not get good results from one or the other, and that doesn't tell us anything at the end of the day. And what we're trying to do is optimize our system so that we get as many patients to take home a healthy baby as we possibly can. That's our ultimate goal, and that's what we're striving to do. Do you need to refresh? And again, Jason Swain did a paper saying, here are some of the potential problems. And as I said, when I started at the beginning of the 90s, we knew our culture systems were compromised. They were nowhere near optimal. But because we did embryo transfer on day 2, much of its inadequacies were hidden. So we didn't culture any longer, so we didn't have time to cause too much chaos. If we went back to those times and said, we should do day 5 transfers, we'd have a nightmare, and a majority of patients wouldn't get a transfer because we were not good enough at the time, and we didn't recognise some of these issues, particularly around VOCs, particularly about evaporation, pH control, and all these sort of things. As I say, they just weren't in our forefront of our minds. We wanted to get patients pregnant at any cost, and we put 3 embryos back in most, and still the majority didn't get pregnant. So we've come a long way. And if you believe, which I truly do, those results that Klaus has presented, we've come a long way from where we were. I always hark back to the original Louise Brown. If it hadn't been for Bob Edwards' stubbornness, Louise Brown was born on their 104th attempt. So not with her parents, of course, but with other parents around the time. If he hadn't truly believed that it would work, most people would have given up by then. So we've come, I say, a significant way along the pathway. So can it be used with time-lapse? Of course, this is what brought about the big expanse in single-step media. So even those that didn't really believe it early on thought that actually if you've got time-lapse and you can observe embryos without disturbing them, then wouldn't it be great if we had a media that could support that development? And so we've seen a growth in single-step media from that point onwards, whereas global for many years was on its own and stood out from the crowd. So again, it can work, it does work, and again, some of the research that's been done will show you can do it uninterrupted. The question, I don't think we need to answer this, does it work as well as sequential media? Then I think the answer is obvious, it does. There's some early papers, and again, they don't show significance in some of the numbers that are relatively low, so the number of patients for pregnancy rate, for implantation rate, but where you had more numbers, i.e. in this case in blastocyst formation, then you do get a significantly increased number, whereas the others are not significant. And that's more or less what we saw across the board initially, and that's what people were looking at. Was it the same, or would it give us the same results? And if so, it was beneficial then to use it because it had some other advantages. If you use a single-step media as opposed to a sequential media, your life suddenly becomes a little bit easier. And again, in labs that are very busy, stressed, anything that can cut down workload, potential to make mistakes is obviously going to be beneficial. So even though these things didn't show particular benefits one against the other, I expect if we looked at utilization rate as well, that would be significantly better. But people moved because if it wasn't any worse, why use 2 media when you can use 1? Makes our QC and everything else that much easier. So just another one that shows exactly the same thing. So just to reiterate, global media works as well as any other. It seems to be one of the most robust media on the market. We've now shown that whether it be LGPS or LP, it works the same. Many of the clinics that used it initially in the UK before the CE marking issue raised its ugly head, didn't use HSA at all. They use dextran and other macromolecules, and it works just as well with those. So it's a very robust media that gives you great results, and like all media, only if you use it optimally. That's what we're here for, the Medical Affairs Department within Cooper Surgical. We've been to some of the labs of people here. It's an open invitation for me to come to your lab if you need it. You know, we can help, and our true goal is to make sure that if you use any products from Cooper Surgical, that you use them to their best advantage, and therefore we get patients, as many of them, taking home healthy babies as possible. Okay, there's a few more slides, but I think I'm going to leave it there. I think that's the message I want to take home. Don't be afraid of the media, don't be afraid of single-step media, but please do take care that you try as far as possible to optimize your conditions. If you do that, then I'm absolutely certain you'll get great results, and we've got a good team around you to help you do that. So thank you for your time, and any questions, please feel free. Only 10 past, that's not bad.
We've been talking about a lot during this day about to refresh or not refresh of the media. And we are always talking about, I think that both Jose and Jorge, they are working with humidity. Is that right? No? It's dry? Okay, so my question is, how important is it if you have a dry incubator to change or not to change? Because this is one of the, at least for my clients, is the first question if you want to move from sequential to unique.
This first raised its head in New York. If anybody's been to New York in the winter, particularly go Christmas shopping in the centre of New York, you get an Arctic blast, and the air becomes extremely dry. So we had a clinic in New York where they'd had great results using single-step media, and then all of a sudden their results started to dip. And when they looked at any possible cause, they realised that the relative humidity in the lab had dropped to 14%. So incredibly dry and below where it should be for people to work in the lab. And they realised then that what they had to do was they had to optimise their system to take that into account. And the two things they had to change were the drop size that they used, because as we said earlier, the smaller the drops you use, the great surface area to volume ratio you have, and therefore more chance of evaporation, and also the type and amount of oil that they were using. So I use the words of Tim Schimmel, who's an embryologist in the US and done a lot of work in this area, and he recommends there's always 3 millimeters of oil above the top of your drop. Okay. Now, when they did that, they switched to the heavier oil. They put more in the dishes, and they changed the volume of the drops. And in their system, then, uninterrupted, they had no problem. It's like everything that we talk about. All of these things that I talk about today, and we've discussed, and Klaus has discussed, they all have to be tested in your environment. And so, if you have any doubt whether it will work or it won't work, you have to do a study side by side to determine it. So you take good prognosis patients, as Klaus did, because there's no point working with the worst ones if you want to prove something will work. So you take your best patients, and you take half, and you change on day 3, and you take the other half and you don't, and then you have a look and see whether there's any benefit or whether there's any advantage. On day 3, you're not changing the media. The media will support development, you're changing the oil. But if you tell embryologists to change the oil, unfortunately in some parts of the world, that's what they'll try and do. Yeah. And it becomes a nightmare, so we just say change the media. But it's really the lab environment and whether it's impacting on the oil. And then that includes humidity. So if the lab becomes very dry, then of course you run more risk. But if you have enough oil and big enough drops, that risk is minimised. But as with everything, you need to test it in your system, in your environment, to see that it works properly.
And we have a test. So, luego tenemos un examen a ver si de verdad os habéis quedado con todo o no. Bueno, pues eso es una pregunta que yo personalmente tenía porque muchas veces me surge esa duda cuando voy a visitar y es algo que es importante saberlo.
Oh, here we go.
When we talk about viscosity in the oil, I don't really understand the importance of working with one light oil or viscous, very dense oil in terms of performance of the culture media, because we have several EV clinics or several clinics where we can have different culture media, same system, and we don't see big differences between...
In most cases, the lab environment is good and there's no extremes, makes no difference. It just means that with heavier oil, so more viscous oil, it takes longer to equilibrate the dishes. But when we're talking about extreme conditions, like in New York, where the air is very dry, then you need to take specific precautions. And so the thick oil or the viscous oil in that environment, it takes longer to equilibrate, but it takes much longer for the gases and any evaporation to occur when the relative humidity in the lab has dropped. So it's like a barrier.
Is there any study that we can look? I mean, it is every study comparing osmolarity of the culture media under these two conditions.
There are studies out there that will show you depending on the oil. So there are studies, very nice studies, that show you about the changes in osmolarity, even going from I think 30 to 50 microliters in a small well. Even just changing that amount of oil had a significant impact. But they don't tend to do the study between different oils. So they do it with the same oil and looking at the amount of oil that you use. Unfortunately, worldwide, the trend is for people to use smaller and smaller drops and less oil to try to save a little bit of money. And ultimately, this is going to cause problems, particularly if you want to do day 5 transfers. In the majority of cases, in the majority of labs around the world, the light oil is perfectly fine. It's only when you're going to have extremes that it seems to make a difference.
Yeah, but to me, I would like to see some sort of graphics.
Yeah, I know.
I mean, maybe in your lab, in Cooper lab, maybe you can do an experiment.
We were just talking.
No.
We were talking about this at lunchtime. We now have some technicians based in Copenhagen that are starting to look at these things. Mm-hmm. So the first thing that they're looking at is changes in osmolality. So we're meeting them, unfortunately it's not till the beginning of December, to give them ideas about what they can do. And this is one of the things that's on the table. It's not just anecdotally talk about
so
it,
you have
but have
real
data.
Absolutely. To show
I can only tell you anecdotally at the moment,
but
we want data. And so they can do that. They've got all the equipment, they've got all the setup to be able to show you which is better and what type of difference it will make. So there are some publications with volume of oil, but not type of oil.
Right,
yeah.
Okay. And another question regarding pH monitoring. I saw in the brochure that Cooper has another way of pH measuring method. And nowadays in the market we can see different approaches, like we can use pH Online or the Epoch or normal pH meter, and the same. Do we have data comparing the same culture situation, the same the same media and the same CO2 and the pH that you can measure with different instruments, with different items? Do we have this data?
Yeah. I've done some of that work, and Tim Schimmel's done some of that work in New Jersey, but it was mainly to validate the blood gas analyzer as a sensible tool. I was in France last week doing some pH testing for a lab there, and they have SafeSense attached to 2 of their incubators, and it kept telling him that the pH was dropping, and so he kept dropping the CO2 level, and at one of the incubators it was at 4%. And I said, well, I'm absolutely certain if I test with my blood gas analyzer that pH will be way too high, which is exactly what it was. So in his scenario, the SafeSense was not giving sensible information. like anything, if you don't calibrate it properly, it won't give you the right, and that's where his problem was, that he'd not set a benchmark for it. So it didn't know, it's a machine, so you have to tell it everything. It's not intuitive. So he had not told it what to start from. So saying it, when I set the CO2 at this level, with this media, I get this pH. And if he'd done that, it would have been giving him sensible results. But I don't know whether anybody's published it. We've done it to validate what we do when we come to see you, to make sure that the blood gas analyzer gives us sensible results. Because if we advise you based on what we find, it has to be real and pertinent. If it's just something we make up, then labs could change what they do and it'd be a disaster. So, we need to make sure it works. But again, I don't know whether it's been published.
Okay, thank you.
I have one question, Steve, on the washing of the oil. Today we heard Klaus giving arguments very convincing about how they wash the oil. What is your take on that?
Yeah, again, there's different oils on the market. Some are not washed at all. Some are washed with the water that the media is made from. Some are made with the culture media, and then some are washed with the media with protein added as well. Again, I've not seen any publications that tell you which is the right way to go. Embryologists instinctively think it should be the media, because if it's going to be next to a media drop, you don't want any leaching into the oil. But media with water, just washed with water, has been sold for years and years and years without any potential or apparent detrimental effect. Now, whether those labs would have got better results if they'd used media washed oil, we'll never know. I do believe it has to be washed with something. There are lots of things in oil. It comes from petroleum. It's a byproduct. It has lots of horrible things in it. But I think testing of the oil is probably as crucial, what you actually are buying and how it's tested. And again, not all oil is the same. Not all. Some companies sell it and it's not washed.
But
They may well advise you to wash it,
Okay.
but they don't wash it themselves.
I thought the majority were already washed.
The majority do, and you have to be careful.
Yeah, I see. Okay.
Thank you for your presentation. We have an interesting conversation with Carolina over there.
Yes.
And I think it will be interesting to share a little of the conversation.
Okay.
Carolina has an incubator with a premixed gas, and they have to compare one medium, that is stage one step, with the live global.
Yeah.
And it's difficult to obtain good pH for both mediums. So what can recommend for people that are in the same situation that Carolina?
Okay. Yep. The flippant answer is, please don't use premixed gas. It gives you much less adaptability. No, if you use premixed gas and that is absolutely set, the concentration that you have to do, then you have to look at things from the opposite perspective. You have to find a media that will work optimally in that setup, rather than try to adjust a setup to accommodate any particular media. So in that lab's hands, if the Sage 1 step is in the right pH range and the one they're testing is not, and you can't adjust it, there's no point trying that other media when it's not optimal, because it's just not. It's not fair to say it only works as well, because if you'd got it into its optimal range, okay, it may only be the same. We'll never know. So I would say in that scenario, you find the media that works best with that setup if you can't change it, whereas normally we try to change the setup to make the media optimal. So we have to look at it from the other point of view. And unfortunately, that sometimes happens. The whole world is never going to be switched from the medias that they use to, or everybody using one media, I doubt, ever, whatever the arguments might be, because many other factors come into play as well. So, and it's a dangerous place to be. If you've got such massive market share, if anything happens, God forbid, the manufacturing plant burns down, we're in a big problem if nobody else is producing any media. So we don't want everybody, but what we want everybody to do is actually use whatever they do optimally, whoever the media is from. And unfortunately, at the moment, I think there's a lot of culture goes on around the world with media that's giving okay results. And embryologists are happy and say, well, if my clinical director's not making me think all the time about how we get any better, then I'll just stick with where I am. And that may or may not be good in terms of a global performance, but it's okay in a local performance, and that's all they're bothered about. And unfortunately, I do see quite a lot of that with just complacency. Well, if it isn't broken, don't fix it. So if we've got a 25% pregnancy rate and my medical director's not giving me any hassle, then why would I want to go any further? So that's my cynical hat on.
Do you have any questions? Any other?
I'll be back.
I don't.
Hello. Talked about when you're using single-step media, the need to refresh or not on day 3.
Yeah.
And I was wondering, do you see a lot of clinics, if they believe it's not necessary to refresh, so they do continual culture until day 5, is there many clinics that don't evaluate the embryos on day 3? They don't see the necessity to take them out of the incubator?
It's an increasing trend. I mean, at one time, embryologists would be extremely cautious about never looking at an embryo, but from FERT check to day 5. But now time-lapse has come along, and you see, it was very interesting that Klaus saw some differences in early cleavage embryos, even in his relatively small study, that had gone by day 5. So is it worth looking for them if they eventually disappear and don't make any difference? So he's found these differences in his study on day 2 and day 3 with compaction and forecell, etc. But by day 5, they seem to have no relevance, and he gets the same pregnancy rate between the two. So whether you change the media or not, I think is a lab decision which needs to be monitored and make sure it's working optimally. But I do now see many labs who've decided, well, if it works in time-lapse, let's just do a FERT check, put the embryos in the incubator, and we basically don't look at them again until day 5.
Is there people here who don't have time-lapse who don't check embryos on day 3? No, it's people tended to be a bit scared of that up until maybe 2 years ago, and no one did it, and then people have started realizing that, you know, the system is there and it works. So we're only giving a disadvantage if we take the embryos out on day 3. So.
The other trend is I do see more and more labs now doing transfers on day 4, which we never did. I mean, when I was trained, I was told that's the one day you couldn't do transfers. And that might be something that changes where Klaus's work comes in, because if that does help you select better embryos on day 2 and day 3, then you can put them back on day 4 and have less of the hassle that you have with blastocyst formation. But again, he's also wanting to do a lot of trophectoderm biopsy, so he needs blastocysts to do that. Am I free?
No.
Oh. Of course.
I have a question just referring to transfer on day 3 or day 5. I know many people that have changed absolutely to day 5. Even with patients that have a very low prognosis, with a very low number of offsites. And yeah. What's your opinion about transferring on day 3?
Yeah. So again, there's never a simple answer. There's two views. There's a scientific view and there's the human view. So the scientific view is that if you believe your culture system does not stop any embryo getting to blastocyst that has the ability, then you should do everybody to day 5 regardless. But most people, and it's clinicians I think more than embryologists, they don't want to tell the patient early that they've got nothing to transfer. So they want to do a day 3 transfer. Now again, we can't ever separate those out, because what we'd like to do is then say, well, if they didn't have the transfer on day 3 and we'd kept their embryo, they'd have had a blastocyst and they'd have got pregnant anyway. So we'll never know. So I actually think, personally, I would want a very robust culture system and I would take everybody through to blastocyst. I think it's very unfair for some to do a day 3 transfer, give those patients 2 weeks of hope when actually they don't have any.
But if you have imagine you have a good quality, you have just a few embryos. I believe in what you said at the beginning. I think we have not a good system. We have
No,
a good system.
That's the big caveat.
But we have.
If
we
we
are
don't
very
have-
far from
Yeah.
the conditions in nature. So I don't think we have a good culture system.
And I think
we have what we can.
And I think if we could get a good marker, a really truly good marker of embryo quality, we would start putting back embryos on day 3 again.
Yeah, I agree
If
with you.
we could.
Yeah, but you need some biomarkers.
But you do. You need
Yeah.
something that's absolutely robust.
I agree with you.
But I think we would go back to doing day 3 transfers if that were the case. You're right. I mean, probably when you get to the implantation rates that we saw in some clinics that are achieving, maybe in that scenario. But until you reach that point, if you believe that your culture system will not compromise any embryo, then I think the fair thing to do is take everybody through. But if you have any doubts at all, then yes, we do a compromise and do day 3.
But I think you have, there is no doubt that you have some problems in you have a stress. And you
do.
have, with ovum donation, it's another history, but not with a woman of 39 years old. The antioxidant difference is nothing to do with a donor. So you are inducing stress, and you can maybe you can improve the results
I
transferring
agree. Yeah.
before.
And if you believe your system makes it worse, then you should put them back earlier.
In my point of view, what I think is that whatever harm we do, because we in our culture system, in the IVF laboratory of the 21st century, maybe on day 3, those embryos has already, ¿cómo se dice? Están tocados de muerte. They are already, that's what I think, that from the beginning, I mean, I think whatever harm we do in the lab is made from the very beginning. So at the oocyte level, we do harm. If the embryo cleaves at day 2, probably, and if our culture system is not good enough, maybe these day 2 embryos are already less viable, and probably by day 3, you are not going to improve much better your pregnancy rates, because probably those embryos are already more less viable, because they have been in the culture system for 3 days. Yeah, all these mitotic errors that we create in the lab. You see.
I think I like very much this discussion, but I think it's just to think about. I think the embryo has to do many different things, and from day 3 on, they have to do very difficult things. I think the very difficult begins on day 3. So if you maintain an embryo on day 3 in the lab in your best conditions. I have been working for 30 years. My lab is nothing to do with the first lab I had. But I think I will never have the conditions I need for a good cultural system in vitro. Because I am culturing in vitro. And an oviduct that's nothing to do with a dish of plastic with no fluids, no growth factors, no integrins, and no many things. So from day 3 on, I injuring the embryo with my system.
Data right now, nowadays, data are showing that implantation rates are higher on day 5 than on day 3. So I think we are not doing in a wrong way when we are contouring our embryos, because if there will be a very, very big change in between day 3 and day 5, implantation rates will decrease. And what we are watching right now is that we are increasing implantation rates when culturing to day 5. So I think you have to be confident with your culture system, with your lab, with your culture media, with everything.
Like it. I mean, the in vitro culture is in vitro culture. I think, like he said at the beginning, in the in vitro culture, it has nothing to do with the in vivo. So you will never have that conditions. And many people, many patients with a very low response, with advanced age, they don't have an antioxidant system prepared to an in vitro culture. Even if you have the best of the medium, even if you control your pH, even if you have the best of the dishes in the market.
But maybe we don't have to think only about cultures, the culture system or the culture media. When you talk to people who work in epigenetics, for example, they give more importance to ovarian stimulation more than the culture system or the culture media. So the whole treatment, not only culture media or culture system, is affecting the viability of our embryos or our results, not only culture system. Probably the stimulation is more and more important than the culture system. Yeah. I don't know, everything is...
¿Quiere otro más?
I wanted to go back again to the composition of the culture media, because you didn't give us a response on that. But no, the thing is that salts are salts, amino acids are amino acids, but we have been talking a lot about HSA, and I think all we know the paper of Dirlund, and we know that the HSA has variations depending on the source of the human. So do you perform some kind of test on the HSA that you are using for every single lot that you are producing, or?
Yeah, but only in the fact that we do the same that we would do with the culture media, so mouse embryo assay, sperm survival assays, toxicity, etc. We don't test them in a live environment, if you like, to see what sort of human blastocyst rates you can get. I don't think anybody does that. You don't need HSA. A strong UK group who had maybe 5 years ago who had the best pregnancy rates in the UK, they use Dextran. So you don't need to use HSA. There's a lot of speculation about what's in HSA, and the same with the HSA plus alpha and beta globulins, but ultimately you don't need them. You can work without them.
Sorry. Or maybe it's time to change
It is.
the source of protein for something that won't have a human gene.
Yeah, you're right. I mean, the one thing we want
to do.
the
embryologist.
No, but you're right.
So I'm proposing that you have to think about
I'm an embryologist as well. What we want to withdraw from everything that we do is variability. So we want to know that everything we use every time is the same. And that's why when people say, should I use pre-supplemented or supplement media myself? The answer from a good lab practice point of view is you should always use supplemented media, because the manufacturer has a much better way of controlling that than you do when you do dilution in your lab. So we want to remove variability as much as possible. At the moment, we haven't got the data to back up what else we might use, because Dextran equally can't get a CE mark. So we are restricted somewhat in what we do in Europe by the regulations. So in other countries, you can use Dextran quite happily. It has 510K approval for the US, etc. But in Europe, we have to rely on CE, and they are very strict on some of the things they will allow us to do. And unfortunately, Dextran, as with other macromolecules, they won't give CE marking for it.
Do you have more questions? No?
Started slow, but nearly had a fight, so that's even better, you know.
Okay, so voy a comentar una cosita. Okay, in Spanish?
Yes, please.
Okay, but and then I translate. No, you keep here. You have to be here. Bueno, lo que quería comentaros es que como comentaba Steve, que usar los medios de Life Global requiere para tener un resultado óptimo que el lab esté a punto. Lo que nosotros queremos proponeros es quien esté interesado en probar los medios, en vez de mandar un vial a ciega y allá quien pueda que mida el pH y quien pueda que no, pues lo que queremos hacer es involucrar a Steve y también al equipo para que quien tenga un interés real en, bueno, probar y luego, si sale todo bien, cambiar de medio, queremos agendar visitas para poder ayudaros a poner a punto el laboratorio. Así que, como todos tenéis un delegado de referencia, pues estaremos encantados de que contestéis con ellos, o bueno, conmigo siempre, pero si queréis, pues con otros delegados, y intentaremos agendarlos para que sea una cosa guiada, porque nos ha ido muy bien. Creemos que hacer los estudios previos, en vez de lanzar el medio tal cual salió en el mercado, es una cosa que a nivel empresa pues se pensó mucho, evidentemente, pero creemos que ha sido la manera, que más vale ir poco a poco y que estéis contentos con los resultados. Entonces, que lo sepáis, que lo queremos hacer así, ¿vale? Muy bien. So you have to come to the clinics to help
Sure.
them to...
Anytime. It's no problem coming to Spain, as I say. Keep the weather good and I'll be here.
Muy bien. Thank you, Steve.
Thank you.
Bueno, pues con esto vamos a dar por cerrado el día. Muchísimas gracias de nuevo por... una fotito, vamos a hacer una foto. Sí, ahora Elena quiere también invitaros al otro workshop que vamos a hacer en Madrid a principios de octubre. Agradeceros a todos de nuevo, puede ser que estéis aquí, que hayáis madrugado. Ya veo que algunos hemos estado bostezando un poco a esta hora. Y nada, gracias y que os lo vais bien a casa. Las preguntas que tengáis, canalizarlas de nuevo a través del equipo.
Hola, bueno, para los que no me conocéis, yo soy Elena, la delegada de la zona centro, y bueno, en relación con el workshop, que espero que os haya gustado, vamos a hacer un curso de control de calidad en Madrid. Vamos a hacerlo en el Hospital Gregorio Marañón. Creo que va muy al hilo porque se hablará también mucho de pH, de qué tenemos que hacer para optimizar los resultados en el laboratorio. Bueno, hay algunas modificaciones que se tienen que hacer, pero hablaremos de la norma UNE, cómo montar un laboratorio, cómo optimizar nuestros resultados en el laboratorio. También hablaremos de witness y de mantenimiento. Creo que estará muy interesante. Si alguno de vosotros quiere asistir, por favor que contacte con su delegado de ventas, porque las plazas son limitadas, y nos pondremos en contacto con vosotros, ¿vale? Habrá un hands-on también por la tarde, que lo mismo, habrá menos plazas. Si estáis interesados, también decidlo, tanto si es para la teoría como para ambas cosas, ¿vale? Y nada más, muchas gracias.